Hello, my name is Brian Boone. I'm one of the surgical oncologists and researchers here at WVU with a primary appointment in the Department of Surgery and an adjunct appointment in microbiology, immunology, and cell biology. My clinical and research focuses on pancreatic cancer, which is one of the deadliest cancers. Our research team focuses on challenges that we see in patients with pancreatic cancer and tries to understand the underlying pathophysiology that promote these challenges and drive the aggressiveness of the disease in order to develop new therapies to treat it. Our main focus is on tumor immunology in the pancreatic tumor microenvironment. Pancreatic cancer microenvironment is one of the most immunosuppressive tumor types, which is a hallmark of this disease. And as a result, it really doesn't respond to checkpoint inhibition or other immunotherapies. Specifically, we're focusing on the role of neutrophil extracellular traps, or NETs, in the pancreatic tumor microenvironment. NETs occur when activated neutrophils release their intracellular contents into the tumor or circulation. And this was initially described as a way for neutrophils to trap and kill bacteria, but has since been identified in cancers, including our work in pancreatic cancer. We have also done additional studies showing that the surgery involved to remove pancreatic tumors further induces this NET process as part of the physiologic wound healing response, but that may further exacerbate the cancer. Ongoing and future projects in the lab are looking at how NETs impact the pathophysiology of pancreatic cancer and contribute to its aggressiveness. We currently have projects ongoing related to NETs and immunosuppression, as well as fibrosis in the tumor microenvironment, with future projects aimed at looking at the role of NETs on tumor metabolism, angiogenesis, including blood vessel density and function, the formation of metastatic disease, and the development of blood clots, which are a common clinical problem for pancreatic cancer patients, and how NETs contribute to hypercoagulability. We're also looking at the interaction between NETs and other cell types, including other immune cells. In addition to studying the pathophysiology of NETs in cancer, we're also developing targeted therapies to try to reverse NETs using a combination of genetic models, as well as small molecule inhibitors and repurposing drugs. Our lab uses a true bends to bedside and back translational research paradigm that allows us to study these processes using resources spanning mouse and human cell lines to animal models, including orthotopic and transgenic spontaneous models of pancreatic cancer, to human tissues and blood, which we get from the operating room, and then all the way to clinical trials where we can translate our bench discoveries into a clinical trial that actually impact patients and their outcomes of their disease. Importantly, once we perform one of these trials, we also take samples from the trial back to the lab in order to perform correlative and mechanistic studies and try to come up with the next treatment. I think that students who do best in my lab are those that are highly motivated, thrive in a team environment, and really enjoy and appreciate graduated levels of responsibility and autonomy and independence. So thank you for your interest and time watching this video, and I look forward to meeting you.